So this is the second video for the unit on the B1, You and Your Genes for OCR 21st Century Science. And the last clip we looked at how you inherit your features from your parents, and we looked specifically how an offspring was either male or female. So we'll look at in this one is the idea of genetic crosses, because it may well be that you've inherited the same colour hair as your father but not your mother, and possibly you may have inherited the eye colour of one parent but not the other. So how is it that some features are inherited from one parent and some features aren't inherited from the other parent? And also you may well have a feature that neither of your parents have but your grandparents have. So we're going to look at the mechanism for how we inherit that genetic information. So, as we know already, you have 23 pairs of chromosomes making a total of 46 chromosomes. So you have 23 pairs of chromosomes. So, one chromosome from your father and one from your mother, in making those 23 pairs to make 46 altogether. So on this pair of chromosomes, there will be a specific area on the chromosome which is dedicated to a particular feature, or there might be a number of areas dedicated to a particular feature. So let's imagine that this is the particular gene for whether your earlobes appear to be attached or unattached. So an unattached earlobe is one that dangles down, and there's my earlobe on the side, or an attached earlobe is one that just tends to blend into the neck. So depending on what chromosomes you inherit, and therefore what genetic information you have, will decide whether you will have attached or unattached earlobes. So in order to do this, we look at a mechanism known as a Punnett Square, and we have to understand about the idea of dominant and recessive alleles. So let's start with dominant. So a dominant allele, an allele is just another name for a version of a gene, versus a recessive allele. Now a dominant allele is one which, if it's in a pair with a recessive allele, it's the one whose feature will be inherited. So you can think of it as the bully, as the one that's stronger, or the one that's more dominant or in charge. So the dominant allele in this case is, we're going to use E, and we use capital E to demonstrate that something is dominant. So dominant alleles are always represented by a capital letter, whereas recessive alleles are always represented by a lowercase letter. Now in this case we're going to say that dominant allele, which is capital E, is for unattached earlobes, and the recessive allele is for attached earlobes. So depending on what the parents have, that will determine what the offspring have. So let's go through an example and use a Punnett Square to determine that. So in this case, the mother has got these two, remember there's always a pair, and the father has got the same. Now then, we call this, when the alleles are different, we call that heterozygous. So when the alleles are different, one dominant, one recessive, then we say they're heterozygous, they are different alleles. So what kind of earlobes does the mother have? Well, she's got one attached, so one unattached and one attached. So because she's got one of each, it comes down to which one is dominant. So the dominant one always wins. So in this case, her earlobes are unattached. And the same with father. He's got the unattached and the attached. The unattached is dominant, so he has unattached earlobes as well. So the genotype is what type of alleles you've got. and he has the genotype big E, little e, and so does mother. The phenotype is the name given to the feature that you get. So phenotype or phenotype, and genotype or genotype, two different keywords. Genotype, what genes you have or what alleles you have. Phenotype, what is the feature that you inherit. So let's try and find out what the offspring will have by drawing a Punnett Square diagram. So in a Punnett Square diagram, we simply write down the alleles for each parent. So this could be the fathers, doesn't matter which way around you do them, and the mothers go at 90 degrees on the side, as so, and then we just draw ourselves a little grid. Now in order to fill in this one, we simply take the two alleles which would cross at this point, which is the big E and the big E. So big E is a possibility. In this one here, it's small e with the big E for mother, and then down here, like that. So those are the four possible combinations of alleles that the offspring might have. So that's either big E, big E, big E, little e, big E, little e, or two little e's. So in this case, the genotype is that they've got two alleles for unattached earlobes. Okay, so when the phenotype, so when the genotype is that they have the same allele, then that's known as homozygous, i.e. the alleles are the same, and they will have unattached earlobes because they have no choice. Both of the alleles are for unattached. In these two cases, these are heterozygous because they're different, and again there's either big E for unattached or little e for attached, but because big E is dominant, then they will be unattached. And then down here you've got little e and little e, which is again homozygous because they're the same, and these must have attached earlobes. So of the four possibilities of the offspring, three of them will have unattached and one of them will have attached. If we just move over to here, the chance of getting unattached earlobes is 3 out of 4, or 75%, and the chance of getting attached earlobes is 1 in 4, or 25%. So typically questions in exams will ask you to calculate the likelihood or the probability of a feature being inherited, and in order to do that you have to make yourself a Punnett square, do the analysis, and then work it out. Remember, there's always four possible combinations, so it'll always be something out of four, and depending on what the parents have, what alleles the parents have will determine what kind of outcome you get.